Hi, I'm Rochelle Crosbie Watson, and today we're going to talk about myostatin inhibition and the role of myostatin in regulating muscle cell growth and its possible applicability to treatments for Duchenne muscular dystrophy. So in this course, we've been talking about many ways to get at the problem of Duchenne muscular dystrophy, and today we're going to focus on how increasing muscle mass and improving muscle regeneration can be a really viable therapy either on its own or in combination with other treatments to address the problem of Duchenne muscular dystrophy. So in skeletal muscle, many elegant studies have been conducted to demonstrate the very important role of a molecule called IGF-1 in improving skeletal muscle regenerative capacity, in creating skeletal muscle hypertrophy, which is basically the increase in skeletal muscle cross-sectional fiber diameter, or the bulk of a skeletal muscle, which increases the strength of the skeletal muscle. IGF-1 can also inhibit fibrosis. So all of these characteristics together improve muscle survival, and you can easily see that those are beneficial activities for skeletal muscle. There have been many elegant studies demonstrating that IGF-1 treatment of mice that have muscular dystrophy improves the symptoms of muscular dystrophy, and that's illustrated here. These are skeletal muscle transverse sections that are stained with hemotoxylin and eosin, and you'll see that there are two sections taken from the TA muscle of a dystrophic mouse, and outlined in white in the dashed line you can see that there's massive necrosis in the dystrophic animals. These sections were taken at day 19 and day 21 after treatment. So compare the untreated animals with the MDX mice that have been treated with IGF-1 for 19 days and treated for IGF-1 for 21 days. I think you can see that there's a vast improvement in the histological profile of the skeletal muscle. We just don't see that massive necrosis that is so evident in the MDX mice. So we know these were some of the basic experiments that were done to really demonstrate that improving the regenerative capacity of muscle, that increasing skeletal muscle hypertrophy and activating those growth pathways are actually very beneficial to Duchenne muscular dystrophy disease. Use of IGF-1 in the clinic has been fraught with some issues. So scientists have been asking and probing for other ways to tinker with these pathways, and we're going to discover one of those alternate influencing pathways in today's lecture. So let's start with a case study. Let's say that you're a physician and you have encountered this boy who's extraordinarily muscular. His cardiac performance seems to be very normal. He has normal motor and mental development, but at 4.5 years of age, he can hold 3 kilogram weight of dumbbells in a horizontally suspended position, and his family pedigree suggests several people who are unusually strong. What does that sound like to you? Take a minute and think about it. So here's a continuation of the case study. This is an example of what we mean by this 4.5-year-old who can hold dumbbells in horizontal suspension. How many 4.5-year-olds do you know who can do that? Here are pictures of that 4.5-year-old. You can see that they're protruding muscles. This is highly unusual. So, if you are this child's physician, what tests would you conduct in order to understand what's going on at the molecular and physiological level with this child who is unusually strong? Take a moment and think about it. So in this slide, you'll see the complex proteins that are involved in regulating skeletal muscle growth, hypertrophy, and regeneration. On the left-hand side, you'll see IGF-1 ligand in blue binding to the IGF-1 receptor in orange. The binding of the ligand to the receptor sets off a whole cascade of reactions that in the end result in increased protein synthesis. And it's that increased protein synthesis that results in the enlargement of the skeletal muscle fiber because there are more myosin and actin filaments, more components of the sarcomere that are stacked to create skeletal muscle bulk. On the right-hand side, you'll see a set of a cascade of reactions that are set off by, we're going to focus on today, myostatin. So myostatin is a negative regulator of skeletal muscle growth. So myostatin, and there are also other ligands like myostatin called GDF11, activin, that can bind to the activin type 2B receptor, which is shown as a receptor in the plasma membrane. The binding of myostatin to its receptor activates SMAD, and the SMAD sets off a cascade of reactions that inhibit skeletal muscle growth. So these two pathways, one that activates skeletal muscle growth and one that inhibits or is a negative regulator of skeletal muscle growth, are present in skeletal muscle to fine-tune the growth and the regeneration of skeletal muscle. So scientists know that activation of skeletal muscle pathways and the improvement of skeletal muscle regeneration is beneficial to Duchenne muscular dystrophy. So the question becomes, what about tinkering with the myostatin pathway and removing myostatin, removing the brake on skeletal muscle growth, which will have the effect of increasing cross-sectional fiber diameter? Would that benefit muscular dystrophy? And we're going to find out the answer to that in today's question. So what I've highlighted for you here in yellow is the myostatin pathway, and that is the focus of our lecture today. So myostatin is part of the TGF-beta ligand superfamily of proteins. We're going to see on the next slide, this is a very large family of proteins, many of which you may have heard of in your other courses. Myostatin is also referred to in the literature as GDF8, which is growth and differentiation factor 8. Shown here is a ribbon diagram of myostatin. Myostatin is expressed as a propeptide and is cleaved proteolytically to generate its active form. Myostatin is binding as a dimer, and once it's bound as a dimer, it then interacts with the receptor which sits in the muscle cell surface. That interaction of myostatin with the activin 2B receptor is what sets off the cascade of reactions inside the cell, which then puts on the brake for skeletal muscle growth. So, as we said before, myostatin is a member of a large superfamily of proteins, and many of these you may have heard of in your other basic science courses. There is GDF8, also called myostatin. So, this case that we saw at the beginning of lecture about a boy with extremely strong muscles has actually been reported recently as well. So Liam Hoekstra is a boy who does not have myostatin. He, at the age of 6 months, was able to walk up and down the stairs. He was doing chin-ups at 1 year of age. He was moving furniture around the living room at 1.5 years. Can you believe that? At 3 years of age, he had 40% more muscle than your average 3-year-old, and at 4 years of age, he was about as strong as a 7-year-old. He was truly remarkable. A picture of Liam is shown here. He's working his dumbbells. He's very strong. And you'll also be able to watch a documentary about the world's strongest toddler, which is very, very interesting. And this documentary will show you that he is quite extraordinary in his skeletal muscle strength and in the size of his skeletal muscles. So the question is, can understanding the role of myostatin, and in these rare cases of myostatin loss, how that improves skeletal muscle function just in normal individuals, and can we apply that to the problem of Duchenne muscular dystrophy? This is a schematic diagram taken from a quadriceps, and what we hope to show you here is that control individuals, this is analysis of MRI information, that control individuals have a certain amount of muscle mass. Individuals that have a loss of myostatin due to a genetic mutation that affects myostatin expression show a tremendous increase in the size of skeletal muscle and in their skeletal muscle mass. The observation of individuals that are very strong, that have myostatin mutations, has long been documented in other animals. For instance, this is a picture of, there are 2 different breeds, and we're going to look at both of them, 2 different breeds of cattle. One is the Belgian Blue, and the other is the Piedmontese, that have mutations in myostatin. These animals are noteworthy for their double-muscled appearance. They're huge, and it's all muscle. And of course, that's very advantageous for the beef industry. Here's another image, which I hope you can really appreciate how these animals are truly extraordinary. And that's all due to the fact that they've lost this brake on skeletal muscle growth. So skeletal muscle grows unchecked. Here's another example of a normal wild-type whippet dog and a myostatin null dog. So sometimes these animals are bred for their strength and for their muscle mass. Here's an example. In this column, you'll see wild-type whippets, whippets that are myostatin heterozygous or haploinsufficient, so they're expressing about only half of the normal levels of myostatin. And then the myostatin null animals also shown. And you'll see that there's a dose dependency with myostatin, that as you remove more myostatin, the animals get larger. So it turns out in terms of function, these are race dogs, that the myostatin hets, or the haploinsufficient animals, far outperform the wild type, but they actually outperform the myostatin nulls, who are so bulky that it's kind of hard for them to get around. So these animals have been observed for quite some time in nature. So scientists then conducted several proof of concept studies. And the first was to replicate those studies that were done in nature, if you will. And they genetically engineered mice that lack myostatin. And you can see a picture of them here. The plus plus represents the normal wild type animal, and the minus minus represents an animal that's lacking the myostatin gene. And you can see that the animal is just much larger. So fine, the animal is larger, but what does that increase in size due to? Well, scientists showed that that increase in size is due to an increase in muscle mass. And the idea that removing the brake on muscle mass is an alternate way to control skeletal muscle growth was really born out of these initial studies. And so scientists said, well, we haven't been able to have much luck using IGF-1 to regulate skeletal muscle growth clinically. What about investigating these other pathways that put the brake on skeletal muscle growth and removing the brake so that we can still have skeletal muscle growth, but using alternate methods? So several experiments were conducted. Now, you should be able to, based on everything that you've learned in this course so far, to really outline the experiments that would be done in order to generate these proof of concept experiments. The conclusions are shown for you here. So normal mice with a genetic ablation, so these are normal mice that are missing myostatin, show improved muscle strength, improved skeletal muscle hypertrophy, which is measured by the cross-sectional fiber diameter. And there's an enhanced fiber type 2 effect. So it really seems that the loss of myostatin particularly affects type 2 myofibers. And scientists also observed enhanced regeneration in mice that lack myostatin. Now, when that same experiment was done on an MDX mouse, or a mouse that has Duchenne muscular dystrophy, so genetically removing myostatin from dystrophic mice, and the question is, does that improve the muscular dystrophy, or does it not change the muscular dystrophy, or does it exacerbate? So what scientists found is that removing myostatin from MDX mice improved muscle strength. It decreased fibrosis. It decreased adiposity. It also improved the sarcolemmal stability. So these were the proof of concept experiments, the preclinical experiments that were done to suggest that myostatin could be a viable approach to addressing the issues with Duchenne muscular dystrophy. So how do we apply these lessons that we learned from nature? How do we apply that to the clinic, to a therapy? It's really difficult right now. There are some emerging technologies that could address the genome, but in absence of those technologies, how can we inhibit myostatin using a clinically based approach? And there are many different ways that you can think about this, and we're going to just touch on a few. So at the end of the day, the interaction of myostatin with its receptor is what sets off the cascade of reactions that put the brake on skeletal muscle growth. And that's what we're trying to inhibit. So our goal here is to inhibit myostatin. And we can do that by blocking the interaction of myostatin with its receptor. And there are many different ways to do that. Think about it. You could create an antibody to myostatin. So that antibody, or a protein that binds to myostatin and takes it out and prevents it from interacting with the ligand. There are many ways to accomplish this, and scientists have been devising and engineering these methodologies and testing them in animal models. So I'm going to show you some of these early experiments that were performed in the dog model of muscular dystrophy. And we're going to highlight the preclinical treatments in dog because the dog model has many advantages over the mouse model of muscular dystrophy. The muscle fiber type in the dog model is quite similar to humans. The fact that the dog model is much more larger makes the experiments much more similar to those that we would apply to humans. And the dog model also enables us to really study the cardiomyopathy. You can imagine that overgrowth of skeletal muscle sounds like a great idea for improving skeletal muscle strength, but that could be a real issue when it comes to cardiac function. So Lee Sweeney and his colleagues used AAV or adenoviral therapies to deliver a dominant negative peptide of myostatin that would have the effect of inhibiting myostatin. And they delivered this peptide via gene therapy approaches to the golden retriever model of muscular dystrophy. And we're going to look at some of the data here. So we're looking now at cross-sectional area. These experiments are done on transverse skeletal muscle cryosections. And we're looking at control or untreated GRMD dogs and treated dogs. And I think you can easily see from all of these experiments in which they've very carefully looked at many different muscles of the EDL, the FDS. They've also looked at the gastrocnemius muscles. And in all cases, the treated dogs have a much greater cross-sectional diameter than the untreated. So that means that the fiber size is much, much larger. The sarcomeres are growing out in bulk. So we know that this treatment, just by looking at this initial experiment, the treatment is having the expected effect. It is inhibiting myostatin. Leading to greater skeletal muscle growth. Interestingly, these experiments were very similar to those done in the mouse. The myostatin inhibition seems to have a greater effect on type 2 fibers relative to type 1 fibers. The skeletal muscle hypertrophy is really nicely illustrated here. These are H&E stained sections. You can look both at 10x and at 40x from control skeletal muscle and treated, i.e. myostatin inhibited skeletal muscle. And you can really see, particularly in the 40x images, that the cross-sectional area is much, much larger after myostatin inhibition. Furthermore, we've discussed one of the hallmarks of Duchenne muscular dystrophy is the sensitivity of the skeletal muscle membrane to contraction-induced injury. So that contraction-induced injury causes the release of skeletal muscle proteins into the blood serum, and we can measure that. So you see in normal dogs, there is very, very little muscle-specific creatine kinase in the blood, but in untreated dystrophic dogs, there is a very high level of creatine kinase in the blood due to extensive membrane damage from the skeletal muscles. In the treated dogs, you can see that there is a reduction in the membrane damage. Is it back to normal levels? Absolutely not. But clearly there's a documentable difference between the control and the myostatin inhibited animals. Furthermore, there was an improvement in the level of fibrosis in the treated dog models. You can see both for the EDL and also the tibialis, there was a reduction in the fibrotic material present in the skeletal muscle. So, in summary, myostatin inhibition increases skeletal muscle hypertrophy. Fast fibers are preferentially affected, although there's no overall shift in the fiber type upon myostatin inhibition. There's a decrease in fibrosis, an improvement in the skeletal muscle membrane damage. The results from these early studies are inconclusive about the effects of myostatin inhibition on the heart, and that will be an important area to focus on in the future. So the conclusion as well from these preclinical studies is that this treatment should be safe for humans. So there are many current developments that are ongoing that are targeted at myostatin inhibition, and many of these are really creative. For instance, there's an entire field focused on creating neutralizing agents. So far, the work has been done on creating antibodies that bind to myostatin and prevent myostatin from interacting with its ligand and setting off the intracellular cascade. There are also propeptides which inhibit myostatin function, as well as other myostatin binding proteins like follistatin that inhibit myostatin function. Another way to approach this issue is to create antibodies that actually bind to the activin type 2B receptor and prevent the myostatin from binding then to the receptor itself. So there are many approaches to myostatin inhibition, and they're being tested both at the preclinical and the clinical level. So, in summary, myostatin inhibition is beneficial for dystrophic skeletal muscle in several different preclinical models. That's promising. There's an increase in force production, a decrease in fibrosis, and there are many efforts to develop myostatin inhibition into a real treatment, either as a standalone therapy or in combination with other therapies. We still need to investigate the myostatin and its effect on the heart. We need to ask other questions too. Are there other members of the TGF-beta family that could be targeted to achieve the same effect as myostatin inhibition? So it's really exciting when we think about the many different ways to address Duchenne muscular dystrophy and to apply our creative energies to developing those methods. I also want to encourage you to use the information that you've learned about the pathways that are involved in the many different cellular events that take place during Duchenne muscular dystrophy pathogenesis to realize that there are multiple different targets and multiple different levels of ways that we can address this disease. And in fact, the end therapy may be a combination or a cocktail of compounds that hits at many of these different pathways. So I hope that you've enjoyed our lesson on myostatin inhibition, and I hope that this encourages you to think more about skeletal muscle growth.